If you are serious about understanding the neurobiology of movement, of motivation, and, habit formation, you just have to confront the basal ganglia.
There's no way around it.
No. This is, I think, arguably the most architecturally intricate collection of deep brain nuclei we have encountered so far. And it silently orchestrates just about every intentional, voluntary decision you make.
It really does.
So our mission today is a proper deep dive. We're using our source material, this incredibly comprehensive neuroanatomy chapter, to really map the architecture, analyze the neurochemistry, and reveal how this system functions, how it works as the brain's ultimate action selection machine.
I think that's the perfect term for it.
Yeah.
And it's truly a masterclass in biological engineering.
Yeah.
I mean, at its heart, the basal ganglia is a collection of subcortical nuclear masses. They're situated deep within the cerebral hemispheres, and they're very closely related to the internal capsule.
Right, that dense, massive highway of white matter fibers carrying information to and from the cortex.
Exactly.
Yeah.
And, historically, the very definition of what even constitutes the basal ganglia has been, well, fluid.
Yeah.
Which I think speaks volumes about our evolving understanding of its complex integrative role.
That shift in definition is probably the first crucial nugget of knowledge for you, the listener. What did we used to think it included, and what does modern functional neuroanatomy tell us now?
Well, traditionally, the term was quite broad.
Yeah.
It included the corpus striatum, which is the caudate, the putamen, and the globus pallidus. But it also looped in the claustrum and even the amygdala.
Okay, so a much wider net.
A much wider net. The modern, and I would say more functional definition, is far more restrictive. It's focused really on the motor loops. So we now generally exclude the claustrum.
Which is that sheet of gray matter whose function we still don't really know, right?
We still don't have a solid grasp on it, no. And critically, we now include two other structures that provide this essential dynamic modulation, the subthalamic nucleus and the substantia nigra.
So the initial aha moment is that the basal ganglia is defined not just by its location, but by its function. It's a tightly coupled, interconnected circuit.
Precisely.
It's a stack of structures involved not just in, coordinating gross motor control and slow intentional behavior, but also, and this is critical, in emotional regulation, cognitive processes, and habit learning.
That's it.
Yeah.
And you can see why the pathologies are so devastating. The rigidity of Parkinson's or the flailing movements of Huntington's, they only make sense when you grasp the full breadth of this integrative power.
You mentioned the amygdala was part of the old definition. That seems telling.
It is. Including the amygdala in that traditional term was a valid recognition that the basal ganglia is closely related to the limbic system.
Okay.
It's not a pure motor execution structure like, say, the cerebellum.
Right.
It's about linking sensory input, motivation, and emotion to the decision of which movement to execute, or even whether to execute one at all.
So if the system breaks down, we see profound deficits in both movement, execution, and in behavioral drive.
Both. They are completely intertwined.
Okay, so that sets our agenda. We're going to spend the first part of this deep dive just meticulously mapping the architecture, starting with the core anatomical complex. Then we'll move into the critical neurochemistry and, most importantly, the two crucial pathways, the direct, or go, pathway, and the indirect stop circuit.
And then the clinical side.
Then the clinical side. We'll investigate what happens when this exquisitely balanced system fails, which will lead us straight into the realities of major movement disorders and modern targeted treatments like deep brain stimulation.
Sounds like a good plan. Let's start with that architectural tour, deep within the cerebral hemispheres. We can focus on the historical core, the corpus striatum.
Okay.
Now, this complex name, as I said, historically included the caudate nucleus, the putamen, and the globus pallidus. But to understand its function, we have to immediately define two key functional groupings based on their connectivity.
And the first of those groupings is the neostriatum, which I think is more often just called the striatum today.
Exactly. People just say striatum. And the neostriatum consists of the phylogenetically newer structures.
Yeah.
That's the caudate nucleus and the putamen. These two are, for all intents and purposes, functionally inseparable.
How so?
Well, they share this remarkable commonality in their cellular makeup, their neurochemistry, and most importantly, their role as the principal input-receiving structure for the entire basal ganglia system.
So this is Grand Central Station, the front door.
It is the massive front door where all cortical information enters.
Okay, then we have the second grouping, the lentiform nucleus. This is where the putamen and the globus pallidus are paired up, but this is more of a visual or spatial thing, isn't it?
It is purely a descriptive spatial term. The lentiform nucleus just describes the lens-shaped complex these two nuclei form. When you look at them in a cross-section, they're sitting deep to the internal capsule.
Understanding its physical boundaries seems really key to getting your bearings in there.
It is. This lens-shaped structure is separated from the cerebral cortex and the nearby claustrum by the external capsule. And that's a thin but crucial layer of white matter.
So you have all these layers of white matter, the internal, external, and even the extreme capsules, sort of walling off these different gray matter structures.
And that shows you just how tightly packed the central brain structures are. It creates these distinct information silos, even though they're millimeters apart.
All right, let's zoom in on the specific anatomy. We have to start with the caudate nucleus. The source material gives a fantastic visual analogy for this. It's a massive, C-shaped, almost tadpole-shaped mass.
It is. It's not a simple sphere. It's a long, winding structure that actually tracks the path of development, and specifically the lateral ventricle.
And that C-shape is vital.
Absolutely vital. It tracks the C shape of the lateral ventricle as it wraps around the deep structures. We can define three distinct parts. First, there's the large rounded head. It occupies the superior and anterior aspect, and it actually bulges into the anterior horn of the lateral ventricle.
And that's the bulk of it.
That's the anatomical bulk, yeah. And it's responsible largely for the executive and cognitive portion of the striatum's function.
Then it arcs back and down from there.
Yes. The structure curves superiorly and then posteriorly, and it thins out into what we call the body. And finally, the structure forms this long, thin, curved tail that extends dramatically backward, loose inferiorly down into the temporal lobe.
Wow, that far.
That far. And it eventually terminates right at or near the amygdala.
Now that is a fascinating connection. That anatomical linkage via the tail is what physically connects this motor processing hub to the centers of fear, emotion, and salience detection.
It's an immediate spatial indicator that your movement selection is constantly being informed by your emotional state. They are physically wired together.
And in its posterior course, the body and tail of the caudate, they maintain a specific, consistent separation from the thalamus, right?
They do. They're kept apart by the stria terminalis, which is a fiber bundle that runs alongside the thalamus, carrying information between the amygdala and the hypothalamus, and also by the thalamostrate vein. This boundary is very consistently maintained throughout the structure's entire winding path.
Okay, let's turn to the putamen. If the caudate is this sweeping C shape, the putamen feels much more dense and more centrally located.
It is. The putamen is the most lateral part of the basal ganglia complex. It sits immediately medial to that external capsule we just mentioned.
And while the caudate and the putamen form the functional unit, the striatum, for most of their length, they're physically separated, aren't they?
They are fiercely separated by the massive anterior limb of the internal capsule. That physical separation tells us they are likely handling different streams of cortical information for different types of motor programs.
Yet they are functionally united, and there's one crucial spot where they're physically connected, which sort of reinforces that whole striatum unity idea.
Yes, and this is a highly significant anatomical detail. It really underscores their shared input role. At their anterior ventral junction, the internal capsule simply doesn't intervene. It doesn't get in the way.
So they fuse together.
The putamen and the head of the caudate nucleus merge seamlessly, and this forms the ventrolateral striatum. This fusion, which you can sometimes see as thin bridges of gray matter passing through the anterior perforated substance, is the physical manifestation of their common neurochemistry and function.
And this is where we find the nucleus accumbens, right?
That's the heart of it. That fusion area contains the nucleus accumbens, which is the absolute core of the motivational circuit.
So we've established a complicated mapping here. The putamen teams up with the globus pallidus to form the lentiform nucleus spatially.
Geographically.
But it teams up with the caudate nucleus to form the striatum functionally. The putamen really is the anatomical and functional crossroads of the entire input system.
It's the hinge of the entire input complex. And understanding that slight anatomical separation, separated posteriorly by the internal capsule, but merging anteriorly, is the critical prerequisite for understanding the dorsal striatum, which is the motor executive bulk, and the ventral striatum, which is that limbic motivational fusion. We'll definitely get into that.
Okay, so let's move from that static map to the dynamic flow. If we agree the basal ganglia is the brain's action filter, then the striatum, the caudate, and putamen together is its grand central station.
Oh, great analogy.
The source material really emphasizes that it is the principal input structure. It receives input from almost the entire cerebral cortex. I mean, that's just staggering when you think about it. Every decision, every sensory impression, every plan, it all funnels through here.
The sheer density is immense. We refer to the volume of incoming fibers from the cortex as the corticostriatal projection. And these inputs are consistently and universally glutamatergic.
So they're excitatory.
Yeah.
Powerful.
And excitatory. The cortex is constantly screaming, do something, or pay attention to this. And the striatum has the enormous job of filtering and integrating that relentless input.
But the filtration isn't haphazard. It's not just a big soup of signals. The incredible volume of input adheres to a very strict organizational principle based on topography.
Meaning specific cortical areas map to specific striatal areas.
Yeah.
Yes, this ensures that the motor loop doesn't get confused with the cognitive loop or the limbic loop. It's all highly organized.
So the vast majority of the striatum forms what we call the dorsal striatum, and this part is dedicated to high-level motor control and executive planning.
That's right, and we can get very specific about the mapping that defines its function.
Okay, so let's start with movement itself, the pure mechanics.
The primary motor and somatosensory cortices, so the areas responsible for the mechanics of movement and registering physical feedback, they project massively and overwhelmingly to the putamen.
So the putamen is the motor hub.
The putamen is the dedicated motor execution hub of the basal ganglia. No question.
And what about the rest of the cortex? The planning, the thinking parts?
So the vast association cortices, that's the frontal lobe for planning, the parietal lobe for spatial awareness, and even the occipital lobe for visual processing related to movement, they project primarily to the caudate nucleus, and also to the more anterior parts of the putamen.
Which means the caudate is primarily the cognitive and executive planning hub.
Exactly. It processes the why and the when before the putamen processes the how.
Okay, so let me see if I have this right. If I decide to pick up my coffee mug, my frontal association cortex sends the pawn to reach signal to the caudate. And the caudate helps integrate that plan, sort of green lights it, before the primary motor cortex sends the specific muscle activation signal to the putamen.
That's a perfect illustration. It's two parallel streams running through the dorsal striatum. They work together, but they maintain these distinct inputs and channels.
Now remember that merger we talked about at the front, the fusion?
Yes, that brings us to the ventral striatum.
The functional link to emotion and motivation.
This area, which encompasses the nucleus accumbens and those merged anterior segments of the caudate and putamen, is completely functionally distinct. It receives highly specialized input.
Not from the motor cortex.
No, it gets input from the limbic system, the hippocampus and amygdala, as well as the prefrontal and olfactory cortices. This is the crucial intersection where emotion, memory, and reward signals are linked directly into that action selection machinery.
It's why habits become so strong.
It's why habits become strong, and it's why drugs of addiction are so reinforcing. They essentially hijack this ventral striatal system.
Okay, so now that we know where all these signals land, we have to understand the cellular architecture that processes them, what's actually happening inside the striatum.
Well, the microanatomy is surprisingly dominated by a single cell type.
Yeah.
The overwhelming majority, we are talking about 95% of all neurons in the striatum, are the medium-sized spiny projection neurons, or MSNs.
And the name itself gives us the key morphology, right? Medium-sized, spiny.
Indeed. They are medium-sized cells, they're incredibly densely packed, and they're characterized by their extensive dendritic fields, about 100 to 400 micrometers wide, which are covered in these dense, mushroom-shaped dendritic spines.
And those spines are where the action happens.
They are the critical postsynaptic sites. It's where they receive the thousands of glutamate terminals from the cortex, and also where they receive the crucial dopaminergic input from the substantia nigra.
These MSNs are the output cells of the striatum, which makes their neurochemistry vital to the whole circuit.
Absolutely, and they are entirely GABAergic.
So, inhibitory.
Yes. This means that despite receiving this massive excitatory input from the cortex, the striatum's own output is almost exclusively inhibitory. This is a fundamental organizational principle of the whole system. Inhibition is used to sculpt the flow of excitation.
And there's another layer of complexity. These MSNs are functionally distinct based on the neuropeptides they co-express.
That's right, they express either substance P or enkephalin. And this is the anatomical pre-sorting mechanism for the direct and indirect pathways that we'll talk about.
So substance P neurons drive the go signal.
And enkephalin neurons drive the stop signal. It's all sorted right at the input stage.
So the cortex shouts, go, with glutamate, and the striatum receives the signal, organizes it, and then sends out a precisely targeted slow down or stop signal using GABA and these neuropeptides. What about the other 5% of cells?
The remaining small population or the large cholinergic interneurons that use acetylcholine or HE.
And they stay within the striatum.
Yes, they're intrinsic interneurons. They don't project out. And they play a critical regulatory role, modulating the activity and the plasticity of those MSNs. The intricate chemical balance, dopamine, GABA, and acetylcholine, is what determines how effectively any given signal passes through the striatum.
This brings us to the chemical powerhouse, dopamine. The source material makes an absolutely astounding point. The striatum contains the highest concentration of dopamine in the entire central nervous system, 5 times higher than any other brain structure.
It tells you something.
It suggests dopamine is the pivotal modulator, not just another simple signaling molecule.
That density is necessary because the striatum is the endpoint of the nigrostriatal dopamine pathway. All of these dopaminergic terminals arrive exclusively from one place, the substantia nigra pars compacta, SNPC, in the midbrain. And the dopamine release here, it regulates the efficacy of all those corticostriatal inputs we talked about. It turns the volume up or down on the cortical signal.
Let's elaborate on the reward loop here, because this is so important for behavior. Why is dopamine critical for learning and addiction?
Dopamine functions as a teaching signal. When an action is followed by a rewarding outcome, say, you eat a delicious piece of cake, the SNPC releases a burst of dopamine in the ventral striatum, the nucleus accumbens.
And that burst reinforces the connection.
It strengthens the specific corticostriatal inputs that led to that successful action. It mediates the reinforcing and rewarding properties that are essential for habit formation.
And which are tragically exploited in addiction.
Tragically. The system is essentially learning which decisions to prioritize for survival and for pleasure. It's a very powerful, primitive learning mechanism.
And the final piece of this striatal puzzle, the mechanism that allows dopamine to be so versatile, is the differential expression of its receptors, D1 versus D2.
This is absolutely fundamental. The MSNs destined for the direct pathway, the go signal, they express D1 dopamine receptors in substance P.
And D1 receptors are excitatory.
Well, they are coupled to G proteins that stimulate cellular activity. So yes, the effect is that dopamine excites this go pathway.
Okay, and conversely?
Conversely, the MSNs destined for the indirect pathway, the stop signal, they express D2 dopamine receptors and enkephalin. And D2 receptors are coupled to G proteins that inhibit cellular activity.
So dopamine inhibits this stop pathway.
Right.
Wait, let's just pause on that because that's a deep insight. The same exact chemical, dopamine, is performing 2 fundamentally opposite functions at the exact same time. It's exciting one population of striatal neurons and inhibiting another just to manage the decision of whether to act or not.
It's an evolutionary masterstroke of efficiency. Dopamine doesn't just send a single message, it tilts the entire balance of the system. By simultaneously promoting the go signal and dampening the stop signal, the net effect of a dopamine release is to powerfully bias the entire system toward action initiation and selection. It makes action more likely.
Okay, so the striatum has now received, filtered, and chemically modulated this massive cortical input. Now it needs to be processed further before it can exit the basal ganglia. This processing happens in 3 crucial intermediate and output hubs. Let's start with the globus pallidus GP.
We can think of the GP as the primary gatekeeper that maintains the system's inhibitory brake. The GP sits just medial to the putamen, and it forms the inner half of that lentiform nucleus we talked about. We must immediately recognize its functional division into 2 parts. The external segment, GP, and the internal segment, GPI. They're separated by a little sheet of fibers called the medial medullary lamina.
And crucially, what kind of neurons are in there?
Crucially, almost all neurons in both segments are GABAergics. So, again, they are inhibitory.
So we have an inhibitory input, the striatum, feeding into inhibitory processing hubs, the pallidus. This seems counterintuitive.
It does, but it's all about disinhibition. The GPI is the essential, final output nucleus of the basal ganglia to the thalamus. Its default state is to maintain a constant tonic inhibitory brake on the thalamus.
It's always firing.
It is always firing. It's always telling the motor thalamus, do not move, do not move. The entire function of the basal ganglia circuitry is to find a way to momentarily silence that GPI output, just for a moment, to allow a specific movement to burst through.
To carry that critical inhibitory message, the GPI fibers need to exit the core complex. The source details 2 distinct bundles of efferent fibers. Why 2? And what are they?
Well, they constitute the final common output pathway. The brain needs to thread these fibers either around or through that massive internal capsule, which is the major dividing line in the brain center.
Right.
So the two bundles are, first, the ansal lenticularis, which are fibers that curve deep and around the internal capsule. And second, the lenticular fasciculus, which are fibers that actually pass through the internal capsule in these fine little bundles.
So it's like two separate exit ramps that lead onto the same major highway.
Precisely. They are two distinct anatomical routes just to bypass the internal capsule obstruction. They then merge near the midline, wrapping around the subthalamic nucleus and another structure called the zona incerta, to form this unified thalamic fasciculus, which is often called field H1 of Forel.
And that's the final bundle.
That's the final bundle. It projects powerfully to the motor thalamic nuclei, primarily the ventral anterior, or VA, and the centromedian, or CM. This is the physical mechanism by which the basal ganglia puts the brakes on the motor cortex.
Okay, let's move to the small structure with the giant excitatory role, the subthalamic nucleus, STN. It's small, deep, and utterly indispensable.
The STN is physically tiny. It's a biconvex, lens-shaped nucleus nestled just below the thalamus and medial to the internal capsule. Its small size completely belies its enormous functional importance.
And what makes it unique in this entire BG circuit, outside of the cortical input itself, is its chemistry.
Its main output is glutamatergic. It is the system's primary excitatory accelerator.
So if the GPI is the inhibitory brake, the STN is the excitatory foot on the gas, but it's directed at that brake.
That's the dynamic tension. The STN receives strong inhibitory input from the GP. That's part of the indirect pathway. But his own projections are highly excitatory, and they target both the GPI and the SNPR.
So it's this tripartite connection. GP controls STN, and STN controls GPI-SNPR.
And that is the central mechanism for tightly regulating the stop signal. The STN is the primary driver that keeps the GPI yelling "stop" at the thalamus.
And the consequence of damaging this small, crucial excitatory hub, the catastrophic demonstration of its role, is a condition called hematobolismus.
It's terrifying. If the STN is destroyed, often by a small vascular lesion, a stroke, you immediately remove its massive, powerful excitatory drive to the GPI and SNPR. The GPI-SNPR becomes severely underactive.
The brake is released.
The tonic inhibitory brake on the thalamus is completely released, and this leads to the spontaneous, large amplitude, violent flinging movements that are characteristic of hematobolismus, usually confined to one side of the body. It is a rapid, dramatic onset of pure disinhibition.
Okay, finally we arrive at the substantia nigra, SN, located deep in the midbrain. This is the structure most famously associated with the chemical input.
Right. The SN is divided into two segments, the pars compacta, SNPC, and the pars reticulata, SNPR. They are functionally very distinct but physically connected.
Let's focus on the SNPC, the compact part first. This is the one everyone's heard of.
The SNPC is defined by the presence of large, melanin-pigmented dopaminergic neurons. They're also known as cell group A9. The neuromelanin is actually what makes this nucleus appear dark in a living brain, hence the name substantia nigra, or black substance.
And these are the cells that die in Parkinson's.
These are the cells. They project exclusively up to the striatum, forming that nigrostriatal pathway we discussed. The progressive selective death of these cells is the central tragedy of Parkinson's disease. They are the source of all the modulatory dopamine for the entire system.
And the SNPR, the reticulata part, is actually the functional auxiliary to the GPI, isn't it?
Precisely. The SNPR contains GABAergic neurons. It receives the exact same inputs as the GPI inhibitory input from the striatum and excitatory input from the STN, and it functions as an auxiliary output structure.
So why have two output structures? Do they project to different places?
They do. While the GPI focuses mainly on the motor thalamus, the SNPR projects to the thalamus, and it also projects significantly to the deep layers of the superior colliculus.
And why is that superior colliculus connection important?
It integrates the basal ganglia into basic orienting reflexes. The superior colliculus controls head and eye movements. The SNPR normally keeps this structure inhibited. So when the basal ganglia selects an action, say you decide to focus your attention on a target to your right, it releases the SNPR inhibition on the superior colliculus, which allows the necessary head and eye movements to orient your gaze toward that stimulus.
So it links the high-level action selection to the low-level sensory processing and gaze stabilization.
That's right. It's a beautifully integrated system.
We have mapped the components. We've defined their input and output chemistry. Now we have to combine them. The basal ganglia's entire purpose is to process these parallel segregated loops connecting the cortex, the basal ganglia, and the thalamus. And the goal is, conceptually at least, simple: allow for the selection and initiation of appropriate actions while ruthlessly suppressing inappropriate or competing actions.
And the whole dynamic hinges on modulating the GPI-SNPR's tonic inhibition on the thalamus. You can visualize the GPI as a gate, or maybe a volume knob, that's set firmly on-off. The circuits are designed to very quickly flip that gate to on for a moment, or to reinforce the volume knob to make sure it stays off.
Okay, let's detail the direct pathway. This is the go signal. This pathway is designed for the rapid facilitation of a desired movement.
It starts with the cortex, sending a strong glutamate or excitatory signal to the striatum. In the striatum, this activates the specific MSNs expressing D1 receptors. Those are the substance P neurons. Now, these MSNs are inhibitory. They're GABAergic, and they project directly to the GPI-SNPR. So when the striatum fires, it sends the blast of GABA to the GPI. Because the GPI is itself an inhibitory structure, inhibiting the inhibitor results in
Disinhibition.
The brake is being intentionally and very quickly lifted, but just for one specific motor action.
And when that brake is lifted,
The GPI's inhibitory hold on the thalamus is momentarily silenced. The thalamus is now free to fire. It sends its own excitatory signal back up to the motor cortex, and this initiates or facilitates the desired movement.
and dopamine's role here is to pour gasoline on that fire.
It powerfully reinforces this entire process
Yeah.
by exciting the D1 receptors on these MSNs, which really pushes the go command through the system.
Okay, now for the marvel of regulatory control, the indirect pathway, this is the stop signal. And this is the pathway designed for deliberate, sophisticated suppression of unwanted or competing movements. It's far more complex. It involves 3 interconnected nuclei between the striatum and the GPI.
It is the regulatory circuit designed for control and braking. It's longer for a reason. It starts with the cortex sending glutamate to the striatum. But this time, the signal activates the MSNs expressing D2 receptors, the enkephalin neurons.
Okay.
Now, these MSNs project via GABA, not to the GPI, but to the adjacent GP, the external globus pallidus. This GABA input inhibits the GP.
So the striatum tells the GP to quiet down.
Correct. But here is the critical point. The GP itself is normally inhibitory onto the next structure in the chain, which is the subthalamic nucleus, STN.
Ah, so if the GP is inhibited by the striatum?
The inhibitory signal it normally sends to the STN is reduced. This means the STN is disinhibited, it's let off its leash, and it becomes hyperactive.
And since the STN is the glutamate, or excitatory accelerator,
It sends a powerful excitatory signal directly to the GPI-SNPR.
and that blast of glutamate from the STN causes the GPI-SNPR to increase its inhibitory output onto the thalamus.
Absolutely. The final result is that the GPI is powerfully activated, its stop signal volume is cranked up, the thalamus output decreases, and the unwanted movement is suppressed.
That pathway seems incredibly long, 4 steps compared to 2 in the direct pathway, and, I don't know, metabolically expensive. Why did the brain evolve such a complex 3-stage circuit just to suppress movement when the go signal is so fast and direct?
That complexity gives the system incredible control and precision. The indirect pathway acts as a crucial negative feedback loop. It ensures that as soon as the cortex initiates one motor plan, all the surrounding or competing movements are quickly and broadly suppressed. It allows for focused action selection.
So if you want to move just your index finger.
You must simultaneously suppress movement in your wrist, your forearm, and all your other fingers. The indirect pathway manages this surrounding suppression, ensuring the movement is focused and deliberate, not a clumsy flail.
And the role of dopamine in this indirect pathway is where the complexity really peaks.
It is. Dopamine binds to the D2 receptors, and those receptors, as we said, are inhibitory. So dopamine inhibits the striatal MSNs that start the indirect pathway.
So by inhibiting the pathway that ultimately increases the GPI's output, dopamine is effectively reducing movement suppression.
Yes. The net effect remains the same as with the direct pathway. Dopamine promotes action, it hits the accelerator D1, and it releases the secondary brake D2 at the same time.
This really highlights why the STN is so critical. It sits at the nexus, converting that complex inhibitory cascade from the GPE into a massive excitatory signal aimed right at the final output gate. It's the essential amplifier for the stop command.
It is the choke point. It's what allows the basal ganglia to exert extreme, fine-tuned control over the GPI output. The STN ensures that if the suppression signal is selected, it is delivered powerfully and immediately.
We must also connect these circuits back to the functional segregation we established in the anatomy section. The source material highlights that these loops run in parallel. They're not all mixed together.
This functional segregation is paramount. We have multiple specialized loops, all operating under the same Gauss-Stop principle.
Let's start with the dorsal striatum loop. This is the classic motor executive loop.
This loop targets the GPI-SNPR, which, as we said, project to the classic motothalamic nuclei, like the ventral anterior, VA, and centromedian, CM. This signal then feeds back to the supplementary motor and premotor cortex, and this is what drives the actual execution of physical movement.
Okay, and then second, we have the ventral striatum loop. This is the limbic motivational circuit.
This is where motivation is selected. It starts with limbic and prefrontal cortical input to the ventral striatum, the nucleus accumbens. Its output targets a different structure, the ventral pallidum, which projects to the limbic thalamic nuclei, such as the mediodorsal MD nucleus.
And what does this loop do?
It integrates mood, emotional significance, and reward information. It essentially determines the behavioral drive behind the motor plan. It's the circuit that decides if you care enough to even perform the action in the first place.
So if I'm learning a complicated piece on the piano, the dorsal loop is handling the precise finger movements.
Yes.
While the ventral loop is handling the sustained motivation I need to practice and the rewarding feeling I get when I finally execute the movement correctly.
Exactly. The systems are tightly coupled, allowing for this incredibly sophisticated, integrated decision-making. The basal ganglia is selecting actions at every level: motor execution, cognitive sequence, and emotional priority.
While the core loops dominate the picture, the basal ganglia's influence also descends down into the brainstem, controlling some essential automatic functions. We need to reserve a moment for the pedunculopontine nucleus, PPN, which you correctly identified as a forgotten player in a lot of the simple diagrams.
It often gets left out, but it's crucial. The PPN is located in the midbrain and is anatomically part of the ascending reticular system. Its function, however, is deeply interwoven with the basal ganglia. It is involved crucially in the automatic axial movements that we all take for granted.
Like what, specifically?
Specifically, gait and posture control, the rhythm of walking, the unconscious adjustments you make to stay balanced.
And what defines its connection and relevance to the GPI?
Well, the PPN contains a mix of both cholinergic and GABAergic neurons. It receives significant inhibitory input from the main basal ganglia output structures, the GPI and the SNPR. This descending input from the core of the basal ganglia directly influences the PPN's activity.
And where does the PPN project to?
The PPN then sends projections to brainstem nuclei that coordinate locomotion, like the reticular formation and the red nucleus, and it even influences the cerebellum.
So the GPI-SNPR complex is regulating fine voluntary movements via the thalamus, and at the same time regulating the rhythm and balance of automatic essential movements via the PPN.
That's the crucial distinction. When the core motor loop is trying to select a precise finger movement, the PPN is making sure the rest of your body maintains a stable posture so you don't fall over.
And this is why the PPN has become increasingly prominent in clinical research.
Absolutely. Evidence shows that the excessive inhibitory output from the GPI and SNPR, especially in advanced Parkinson's disease, really interferes with PPN signaling.
And this interference likely explains the symptoms that levodopa, the main Parkinson's drug, struggles to treat.
Precisely. PPN dysfunction is now strongly linked to the severe gait instability, the freezing of gait, and the postural problems. We call them the axial symptoms. They're often refractory to standard dopamine replacement therapy. Understanding the PPN as a descending target of the basal ganglia has opened up an entirely new avenue for therapeutic intervention, specifically using deep brain stimulation.
Okay, let's transition now to the clinical side, where the functional failures of these circuits really manifest in people's lives. The prototype of basal ganglia dysfunction is, of course, Parkinson's disease, PD. This is the disorder of hypokinesia, or too little movement.
It is.
Yeah.
Clinically, PD is defined by what we call the akinetic rigid syndrome,
And that includes bradykinesia.
which is a poverty and slowness of movement, muscle rigidity, the classic resting tremor, which is often what people notice first, and, as we just discussed, severely impaired postural reflexes.
The underlying pathology is singular and catastrophic.
It is. It's the massive progressive degeneration of the dopaminergic neurons in the substantia nigra pars compacta, SNPC. And symptoms generally don't even become apparent until about 60 to 80% of these dopamine-producing neurons have already died, leading to a severe dopamine depletion in the striatum.
Let's trace the circuit failure caused by this dopamine depletion using our Gostop framework. What happens to the direct pathway, the go signal?
In a dopamine-depleted striatum, there's simply not enough stimulation of the D1 receptors. And since D1 normally excites the GO pathway, the direct pathway becomes severely underactive. The striatum can no longer effectively inhibit the GPI. The brake is not being lifted.
Okay, and what about the indirect pathway, the stop signal?
This pathway, which relies on D2 receptors, is normally inhibited by dopamine. With dopamine gone, the D2 receptors are ineffective, and the indirect pathway becomes massively overactive.
So the pathway that increases suppression is running wild.
It's running wild. The GPP is inhibited, which disinhibits the STN, which then blasts the GPI-SNPR with powerful glutamate excitation.
So the GPI-SNPR output is under-inhibited by the direct path and overexcited by the indirect path. Both roads lead to the same functional disaster.
Exactly. The output structures, the GPI and SNPR, receive drastically increased excitatory signals. The result is excessive chronic inhibition of the thalamus. The motor thalamus cannot escape this powerful subcortical brake, and thus it cannot send the necessary excitatory drive back up to the motor cortex.
The patient is unable to initiate or execute voluntary movement. This system is locked down.
That's the definition of akinesia and bradykinesia. The motor system is just locked down.
That detailed circuit explanation is why this knowledge is so powerful. It translates this abstract disease into specific, targetable problems.
But as you pointed out, PD is far more than just a motor disorder. We have to give adequate depth to the non-motor symptoms.
We do. The non-motor symptoms, which often precede the motor symptoms by years and severely impact quality of life, include things like cognitive impairment, depression, anxiety, apathy, and sleep disorders. These are not merely secondary effects of having a motor disease. They stem from the degeneration of other crucial brainstem nuclei.
Specifically, the loss of norepinephrine and serotonin.
Yes. While the SNPC is the primary casualty, PD also involves the degeneration of the locus coeruleus, which is the main source of norepinephrine, and the Raphe nuclei, the main source of serotonin, 5-HT.
And what does losing those do? Well, norepinephrine is critical for arousal, attention, and mood regulation. Its loss contributes heavily to the depression and cognitive slowing we see in PD, and serotonin regulates mood and sleep cycles, and its depletion contributes to anxiety and the devastating REM sleep behavioral disorder, RBD, where patients physically act out their dreams.
That connection to the limbic loop we discussed earlier is also key to understanding some of the behavioral side effects of treatment, right? Like impulse control disorders.
A huge problem. When we treat PD with L-Dopa or dopamine agonists, we are stimulating the entire dopamine system, including the mesolimbic pathway in the ventral striatum. In a denervated and often hypersensitive system, this stimulation can lead to dopamine-related behavioral dysregulation.
Specifically, impulse control disorders, or ICDs.
Right. Things like compulsive gambling, hypersexuality, obsessive shopping, or hobbies. It is a powerful example of how the limbic motivational basal ganglia circuit can be pathologically amplified by medication that's intended for the motor circuit.
Okay, so if Parkinson's disease is the tragedy of excessive braking hypokinesia, then its opposite disorders, the dyskinesias and dystonias, are the tragedy of lost control or hyperkinesia. Too much movement.
The core mechanism of hyperkinesia is a fundamental loss of the inhibitory control exerted by the basal ganglia. It releases the thalamus from its necessary brake and leads to excessive, uncontrolled cortical drive. The thalamus is just let off the leash.
And our prototype for this loss of inhibition is Huntington's disease, HD. It's genetically driven, but the pathology lands squarely in the basal ganglia.
HD is caused by an expansion of a CAG repeat in the Huntington gene. The critical pathology is the severe progressive atrophy of the striatum. And critically, this atrophy selectively targets the MSNs expressing D2 receptors.
The very neurons that initiate the indirect stop pathway. You selectively kill the neurons responsible for putting on the brake.
Exactly. The loss of these inhibitory D2 MSNs results in a massive underactivity of the indirect pathway. The GPP is no longer inhibited, so it becomes hyperactive, and it strongly inhibits the STN. With the STN silenced, the powerful excitatory input to the GPI-SNPR completely collapses.
The GPI brake is catastrophically disabled.
The GPI-SNPR inhibition of the thalamus is severely reduced. This leads to excessive, uncontrolled thalamic output back to the motor cortex. This uncontrolled excitation generates the severe, involuntary dance-like movements we call chorea, which is the hallmark of Huntington's. The basal ganglia simply fails to suppress any competing movements.
That symmetry is incredible. Parkinson's is overactivity of the indirect path. Huntington's is underactivity of the indirect path.
And we see that same principle of disinhibition in other hyperkinetic disorders. Dystonia, for instance. It's characterized by sustained, powerful, and often painful muscle contractions that lead to twisting or abnormal posturing.
So not a flowing movement like chorea, but getting stuck.
Right. While it's often less catastrophic than chorea, it represents a failure of fine-tuned action selection. A posture is selected, but the system fails to inhibit and switch away from that posture. It's often linked to abnormal firing patterns in the pallidum and the STN.
And we must revisit hemibilismus as the most dramatic, acute example of hyperkinesia.
Hemibilismus, with its violent flinging proximal movements, is the result of an acute lesion, typically a stroke, right in the subthalamic nucleus, STN. The destruction of the STN removes the only major excitatory driver to the GPI-SMPR. Without that powerful glutamatergic drive, the GPI-SMPR inhibition immediately collapses, releasing the thalamus from all control. It is pure, massive disinhibition in action.
So our deep dive into these circuits has really provided the roadmap for modern intervention. We can now precisely target the failing component of the circuit.
We can. For Parkinson's, everything begins with the pharmacological treatments, which are dominated by the levodopa era. L-Dopa remains the most effective symptomatic treatment. It's a precursor that can cross the blood-brain barrier, and it replenishes the depleted dopamine stores in the striatum. It dramatically improves rigidity and bradykinesia.
It works by rebalancing the Gauss-stop signal.
It boosts the direct pathway and reduces the overactivity of the indirect pathway, yes.
But long-term use, often after 5 to 10 years, introduces very significant side effects.
The major limitations are the wearing-off phenomena, where the drug effect rapidly diminishes between doses, and the development of severe drug-induced dyskinesias. These are involuntary writhing and rocking movements.
And why does that happen?
It's believed to be caused by the intermittent pulsatile delivery of L-Dopa to a heavily denervated striatum. The remaining dopamine receptors become highly hypersensitive. They react violently to each dose and throw the system too far toward the hyperkinetic, Huntington's-like state.
Before modern pharmacology and electronics, surgeons took a more direct, destructive approach, using historical surgical approaches to correct the failed balance.
Indeed. Earlier attempts included salomotomy, which was making lesions in the motor thalamus. It reduced tremor, but did little for bradykinesia. The more targeted approach, based on our circuit knowledge, was the pallidotomy.
Why target the globus pallidus internus, GPI?
Because the GPI is the final excessively active output gate in Parkinson's. By placing a selective lesion in the GPI, a pallidotomy physically reduces that excessive inhibitory output to the thalamus. This lesion effectively corrects the circuit imbalance.
And it was effective.
Very effective for treating rigidity, bradykinesia, and significantly, it also reduced the severity of those troublesome L-Dopa-induced dyskinesias.
But these were permanent, irreversible procedures. The true paradigm shift came with the development of deep brain stimulation, or DBS.
DBS, which was widely adopted in the 1990s, involves implanting electrodes to deliver high-frequency electrical stimulation. Its critical advantage is that it produces the therapeutic effect of a surgical lesion, but it is entirely reversible and adjustable.
Which means the treatment can be refined over time as the disease progresses or the symptoms change.
Exactly.
The precision required here is phenomenal, given the target nuclei are just millimeters deep in the brain. Which structures are the favored targets? Let's start with target 1, the subthalamic nucleus, STN.
The STN is highly favored because stimulating it at high frequency functionally silences it. It effectively blocks its massive excitatory drive to the GPI. We achieve the functional effect of destroying the STN-reducing GPI inhibition, but without the associated risk of causing hemibilismus.
So by silencing the STN, we release the thalamus from the PD-induced excessive brake, thus enabling movement.
And because the STN is so upstream in that indirect pathway, DBS targeting the STN is highly effective for treating the full spectrum of PD symptoms, tremor, rigidity, and bradykinesia. A major benefit is that it allows patients to significantly reduce their daily LDOPA dose, which mitigates the risk of future dyskinesias.
What about target 2, the globus pallidus internus?
GP. Stimulating the GPI directly achieves the same outcome as a pallidotomy. High-frequency stimulation disrupts the abnormal firing patterns of the GPI neurons, functionally blocking the excessive tonic inhibition being sent to the thalamus.
When would you choose GPI over STN?
GPI stimulation is highly effective for tremor. and is often the preferred choice for patients whose most debilitating issue is severe levodopa-induced dyskinesia, as it directly blocks that dysfunctional output pathway.
And finally, the recent advances tie back to the PPN, our forgotten player.
As we've understood, the DBS of the STN and GPI often fails to completely resolve the complex axial symptoms, the focus has expanded. Researchers are actively exploring and utilizing stimulation of the pedunculopontine nucleus, PPN.
And the goal there?
DBS, targeting the PPN or the caudal connections of the basal ganglia to it, aims specifically to improve gait and postural instability. It's addressing the automatic motor functions that resist dopaminergic drug therapy. This shows the depth of our understanding now, moving beyond the main motor loops to the coordinated brainstem systems.
We have completed an exhaustive map today. We moved from the dense anatomical mapping of nuclei, the C-shaped caudate, the central putamen, the gatekeeping pallidus, the excitatory STN, and the chemical factory of the substantia nigra, to seeing them as a single, tightly regulated, feedback-driven circuit.
And the core functional knowledge, the thing to really take away, is that the basal ganglia's role is precise balance. It's the constant, exquisite tension between the direct pathway, that fast D1-driven go signal, and the indirect pathway, the complex D2-driven stop signal.
A balance managed by the pivotal roles of dopamine, which biases the system toward action.
And glutamate from the STN, which amplifies suppression.
And this knowledge transforms disease. Parkinson's and Huntington's cease to be vague disorders and become specific circuit failures. One, a failure of disinhibition, the other, a failure of inhibition. The modern ability to precisely target and surgically adjust this balance using techniques like deep brain stimulation is a direct result of meticulously mapping these deep structures.
It is. And if we connect this detailed picture to the bigger picture, the basal ganglia is often called the action selection machine. We know that the ventral loop handles motivation, reward, and the formation of habits, connecting our emotional salience directly into our motor execution. Given that every decision, every learned habit, and every motor impulse is filtered and selected by these deep, highly regulated subcortical feedback loops, often far, far beneath our conscious control, it raises a powerful, final question for you to consider.
Which is?
What does this precise physiological governance of action selection in the basal ganglia imply about the true extent of our free will versus the irresistible pull of our deepest, most established physiological impulses? The circuits, you see, are constantly choosing for us.